Scott, that's the edge of the water, I think.
Yeah, that is the edge of the water.
But they're debris. Ooh, there went one. I think they're swimming around the edge. Oh, there's something cool.
You got more than Paramecium in there?
Yeah. It's little round things with like a barrel with a mouth.
Right.
See them?
You have to adjust the lighting a little bit.
Until some swim in. I'm the robotic, I'm a PMCA system on Microsoft.
Don't forget to issue a command, Scott.
A command?
An ESGV.
ESGD command. Focus.
That's right.
Focus.
That's right. It's fun. It's fun to learn how to use these things.
They are swimming along the edge.
Mm-hmm.
Oh, that was a great picture if we could have gotten.
Book Davies is on. They're talking about this electric storm that's out there.
His real name is Steve.
Huh?
Book's real name is Steve.
Steve? Oh, okay.
Davies.
Pekka says, good afternoon, ladies and gentlemen. Happy Friday, or any day is good for me. That's good.
So do you have me so you can, am I in the stream right now?
Yeah, you
Okay.
can see you. Yeah. Not yet. Not yet.
No,
We're not streaming anything
Yeah,
yet.
but I didn't know if I was, because you stopped sharing a while ago.
Yeah, I'll have you reshare your screen.
When you're ready.
Yeah, when you're,
Okay.
yeah.
We're gonna have to get a bird book.
A Paramecium book.
Just a small thing.
Right.
I mean, it's a bird book for small things, so to speak.
Tiny.
They're flying. They're like birds.
I know.
They're flying in water.
Right.
Air is technically a fluid.
I can call me today as IDA advocate for Sweden.
Yeah,
So
that's
happy
important.
for that and getting started to do changes over here. It's great.
Do you want this bulletin board to be visible in the background?
It's not.
Okay. I wish there was a button I could push to take a picture,
There is.
but something I could be ready, like a button on a keyboard I could get. I guess it's snap over here, isn't it?
Astrobiology is a very interdisciplinary field. It's very exciting because it looks at life at the limits and tries to understand whether life could exist elsewhere. Astrobiologists are also trying to find new types of microbes that could be resistant to space extremes. Can we find new microbes that would be useful in space exploration and settlement? And are we the only example of life in the universe, or are there other planets with life? These are fundamental questions, but they also have very practical implications. By learning about life in extremes, we learn about the interaction of life with our own planetary biosphere. That might tell us about the human impact on the biosphere in the future, and the future of life on Earth, or by using the International Space Station to look at the limits of radiation stress in microbes. We might learn something about whether microbes could grow under the radiation environment of Mars, for example, or in other planetary bodies. There's a couple of experiments we've done. One experiment is to expose microbes to space conditions. This is a European experiment called the EXPOSE experiment, where we take microbes onto the space station, we bolt them onto the outside of the station and leave them there for a year and a half, 2 years, to see which ones can survive the intense conditions in space. Over a quarter of the world's copper and gold is today extracted from rocks using microorganisms. It's called biological mining or biomining. And the question is, could we use microbes elsewhere in space to try and extract elements from rocks? The bio-rock experiment was motivated by trying to understand how microbes interact with rocks. Now, you might think it's a very strange thing to have an interest in, but it turns out this is very important. For example, we could use rocks on the Moon, Mars, or asteroids to supply microbes in life support systems that might be producing oxygen or other useful elements and compounds for human existence. We also want to understand how microbes behave inside, for example, filter systems that are circulating fluids and liquids, drinking water. If you're relying on microbes to produce oxygen, for example, for your astronauts, you don't want all the microbes to suddenly die. So you need reliability. So despite the fact they can survive in extremes, we still need to understand how they behave in space and how we can optimize their chances of surviving in space conditions. If I had any wish in space exploration, it would be to try and grow microbes on the Moon or Mars and try and do the real experiments. We can do these experiments on the International Space Station, but these experiments are partly to prepare humans to go to asteroids, the Moon or Mars. So the ideal experiment would be to actually go to these places and grow microbes in the environments in which we actually want to use them.
Hey everybody, this is Scott Roberts with Explore Scientific, and I'm here with Kent Marts. He's behind me here, and he's with the infinity microscope that we have, and we're going to show you some microorganisms that we have bought and some we have collected. But one of the things that I really was concentrating on recently is how did Leeuwenhoek make his microscope lenses. And so I've ordered, I don't have it yet, but I've ordered some glass rods and a Bunsen burner. And what I'm going to do is show you how he did it. And basically what he did is he would take these glass rods, heat it up, and then pull it apart until he had it made like this fine whisker, okay? And then he would have two whiskers on these rods. And then what he would do is he would use a nice technique where he would take the end of the whisker and dive it into the flame, and it would make his lens. Okay, so that's a very interesting aspect. But what he would end up getting is almost a perfectly round piece of glass. And then he would sandwich that between two metal plates, and then he had a nice little holder. And his whole microscope was tiny. It was like that long, that wide, thin, and he would hold it up to his eye with the specimen in there. But, he's the one who discovered so many microorganisms, and he was quite good at making those kinds of lenses. When I look at compound objectives made today, this one's a plan, so that means it gives a nice flat field. It's a 10 power, and then there is the aperture of this lens is listed. The objective lens has several elements, but if you just hold it up to your eye and maybe put your finger underneath it, you can see your fingerprints highly magnified. Now, once we have a very powerful lens like that, we're able to use another lens that would act as an eyepiece, which would increase the magnification even more. If we don't have an eyepiece up there and we just put up the camera, that also creates its own kind of field or magnification. So right now we're going to switch over to Kent. I'm not sure what, Kent, what did you put in your scope? Is
We've got Paramecium
Paramecium
and amoeba. But
and amoeba.
Haven't seen any amoeba.
Haven't seen an amoeba yet. Okay, all right. So we're going to have you go ahead and share your screen.
Yeah, just do that.
There we go.
So what we're doing right now is,
I don't know if they can hear you.
okay, tell them what we're doing right now. is
Okay,
at the
well, you
edge
can
of
see a paramecium has floated past right now. So what he's done is he's just in a drop of water, okay, with paramecium specimens. He's just, he's put it into, on this microscope slide. I think you're at 100 magnification right now.
Right.
And that gives good image scale for the paramecium. There's other things swimming around, though. You can see these little kind of spherical things here. There might be, if he looks around on the slide, we might see some euglena, but it's
The Paramecium seem to be running the edge of the drop of water.
okay. Yeah, so that gray edge is the edge of the.
There we go. There's one.
Yeah, they're trying to figure out how to get out of there, I think. So.
All right, so let's move around a little bit.
All right, so he's going to move.
And this is just, all sorts of stuff moving.
Yeah, there's all kinds of little things in there. Paramecium are also called ciliates, and they are, you can find them in freshwater, brackish, and also marine environments. I guess there's thousands of species of these things. The name Paramecium was coined in 1752 by a guy named John Hill, who applied the name generally to animalcules which have no visible limbs or tails. Okay, so a very, very simple, micro organism. It's only, these things range from 50 to about 300 micrometers. I know that they are kind of predatorial. Okay, I watched them take down a tardigrade. We've talked about that before. But let's see how they're described. It says the body of the cell is enclosed by a stiff but elastic structure called the pellicle. This consists of an outer cell membrane, a layer of flattened membrane-bound sacs called alveoli, and an inner membrane called the epiplasm. The pellicle is not smooth, but textured with hexagonal or rectangular depressions. Each of these polygons is perforated by a central aperture through which a single cilium projects. Okay, so the cilium is kind of like the hair-like edges that you see that I think help move around. There is an anal pore. There is, in all species, it says there's a deep oral groove running from the anterior of the cell to its midpoint. This is lined with an inconspicuous cilia which beat continuously in order to draw food inside of the cell. I mean, it's basically a single cell. But these things can move, and they can use a kind of a whiplash type motion and scoot across the screen, as you can see. So it looks like that's some leftover remains of some other.
Cyclops or something.
poor thing that didn't quite make it, but oh, there you go. Now we don't have a cover slip over this drop of water, so he's having to focus through the water itself.
I'm looking for a Paramoeba, but I don't think I got any.
You'll know the amoeba because it looks like jelly. There's a Paramecium. There you go.
They're running the edge, clearly.
Yeah, there's another one.
That was Ralph. The first one was Mary.
Ralph and Mary, the Paramecium family. They are cool. And earlier I was looking at some rotifers, so, Kent and I may have to make videos or something to, switch scenes easier. But we're showing it to you live, so this is what it's like.
This is science.
This is science.
I wonder if that one's reproducing. It looks like.
I don't know.
Or they're hooked together, maybe. Those look like 3 of them.
Yeah, they have to reproduce. Sometimes they do it all by themselves.
So this we just sit here and watch this.
It's kind of mesmerizing.
Looks like they're hooking together. Look, that one just came up and hooked on.
Yeah. So they're connecting somehow.
Yeah.
For whatever
Looks
reason.
like they have like a barrel with a flowered sort of mouth or something.
Mm-hmm. Rotifers look like that too. They have kind of this mouth that looks like it's spinning or rotating or something, and it's meant to draw food in. That's an amoeba. There you go. That's one, kind of horizontal there.
Let's see what we can do with the light.
Yeah, you can see kind of the edges of it a little bit. These are slow-moving organisms.
Because their body changes shape.
Yeah, and that's how they catch food too. The animal that just kind of hangs out in there, doesn't even realize, oh yeah, this is fine. This thing's not moving fast, and it just step by step kind of comes around them, and pretty soon they're trapped within it, and then they digest this
I
thing.
don't see it moving.
Yeah, I think some of the amoeba videos that I've seen have been sped up many times, maybe like 200 or 300 times. So, not very quick moving.
So we've talked, I was talking about me suggesting that we refrigerate the Paramecium.
Kent wants to refrigerate the Paramecium to slow him down. I don't know if that's ethical. So,
We don't want to freeze them to death. What's that?
but says amoeba look like a clear shadow till you get it focused, and then they're still quite clear, and they're hard to see. He says they move like flagellates. Might be euglena. There's too small to tell. I think there is some euglena in there.
Oh, that's too much. That one,
And if you want, you can change the slide and grab the bottle of euglena there.
I was trying to zoom in on that, but I'm afraid we're going to hit the slide, Yeah, I'm not going to do it.
Yeah.
I just got it wet.
The objective lens is wet now. Yeah, we'll have to clean it. Good thing we know where to get those cleaned.
So I'm just going to lick this one dry.
Jeff wants tardigrades.
Okay, new slide. What do you want to go to?
Jeff would like to see some tardigrades. I do have a jar there. Might still have some tardigrades in it. It's the big jar. You can see if you can find some. We also got some specimens of copepods, which are, someone had mentioned the cyclops organism. And those guys move very quick too. They are they're interesting, but they look like these shrimp or something. They're crustaceans, you know, and a lot of animals survive off of them. So it's pretty interesting.
They probably taste like shrimp.
Probably tastes like shrimp.
Okay, so let's put the lid back on before
Okay.
we spill them. Good lab practices, always. And we put the lids on loose.
Yeah, that's right.
So I'm gonna find the edge of the drop.
Usually you'll find tardigrades on a piece of moss or something in there. Harold says, how does your own blood look under the scope, or is that a good subject to study?
I'll bleed for science.
You'll bleed for science? Yeah.
For science.
We'll get some of Kent's blood next time.
Sure. Oh, tastes like iron.
He's got lots of iron in his blood. Eat spinach every day. Book says blood is fun. It's pretty interesting, the video that we watched, though, about how they're actually exposing microorganisms to the environs of space. I wonder how many of them survived. I know that tardigrades have survived. Not all of them, you know, they put a bunch of them out there, but a good percentage did survive the vacuum of space.
And the radiation of space.
Mm-hmm.
There's not much in this drop of water moving around.
What is that? We may get skunked.
And there's nothing moving in this stuff.
Oh, go to the left. What's that? Might be a tardigrade exoskeleton.
There's something moving in there, right?
You want to share your screen again?
Have I not?
Yeah, share your screen again.
Something moving right there in the dead center. And this is so sensitive. If you're watching your screen.
Okay, so roll the mouse thing and zoom up on it, on your mouse. There you go. That.
Right here. 'Cause there's something right there moving through. There's so much vibration. I mean, basically, I move my feet.
Yeah. Oh, sure. That's a microscope.
And the floor shakes.
A concrete floor shakes.
The concrete floor shakes. Okay, so I'm going to switch to another slide. Or
Okay.
another-
All right.
Try and suck up something else and see what we do. 'Cause we're trying to get some algae going deep.
Martin Eastburn says, put a pinch of grass into water and wait a day or two, and you'll have a farm of all sorts. Ollie says, yeah, I saw the palisade cells and the phlegm and the xylem cells. Ollie says, I also saw a wasp under a microscope once, and its stinger was beating after death. Jeez. I see some. I see it. It looks like amoeba.
I got a big honking huge piece of,
Something. All right, share your screen again so they can see.
I got a gigantic piece of, moss in here. I'm just gonna keep searching for it until I find it.
Oh, okay.
There's nothing there. Oh, here we go.
Okay.
Too much.
Now you might want to increase the light intensity now,
Yeah.
because a lot of times tardigrades hide in this stuff. There you go. Get a focus. And kind of search around and see if you can find one.
That wobbling looking stuff is the actual surface of the water.
Yeah, it is the surface of the water.
Just like the atmosphere boils for us astronomers.
Mm-hmm.
It has to be a tardigrade. There's
That's something. Yeah, I see it. Now increase the light intensity more.
That's all the way up.
Okay. And maybe center it up. He might be dormant.
Could be dormant or a shell.
Uh-huh. It's really clear. It does look like a shell.
It's not moving.
And that's something too. See that off to the lower left-hand corner? No. Come back down. Right there. All the way to the left.
The dark thing?
Not the dark thing, the thing that's to the left of the dark thing. That thing. I don't know if that's just,
Could be a twig.
not sure.
There's not a lot moving in this.
Well, they could have gone dormant. It's possible. Well, by next week, I hope to have the glass stems and the Bunsen burner. I want to figure out how to make a microscope lens for myself. I think it'll be a lot of fun.
And basically, like I was talking about, it's just two convex lenses glued together.
Right. Well, okay. I think that we have seen some things. We got kind of skunked. We learned more about astrobiology today. We will be back with more of these programs next week. But tonight we have Terry Mann and the Astronomical League and Howard Eskeldson coming on in about an hour and a half on the Astronomical League Live number 5 right here on the channels that you're watching on right now. So I hope that you join us and show the Astronomical League some support. And Kent and I will figure out more about the mysteries of microscopy. And thanks for watching. We'll see you in a little bit. We're learning.
There's nothing alive in this stuff.
This is interesting, though.
It is interesting. Just looking at algae that's green, it shows up red here.
Y'all have a good weekend.
You're still alive?
Oh, yeah, we're still alive.
Okay, so let's pick something else here.
Oh my God, look at this thing. He's about to devour this little microorganism. Don't.
We're gonna get some Euglena. Okay, there's gonna be life in 2 drops of water.
This organism is kind of hooked to the, it anchors itself, and you can see it's got these real fine hairs around its mouth. That's how it detects things. And yeah, it just captured something. Done.
Okay, so there's the edge of the water. Ooh, there's some life in this.
Want to show it?
Yep.
Kent thinks he has something here.
Don't know what it is. We need a bird book.
Oh, these are Euglena. I know that.
We need a bird book.
You need a bird book. You need a Euglena book.
No. That's Euglena?
That's Euglena.
What do these things do?
There's single cell flagellate, and they're best known and widely studied member of the class Euglenoidea, diverse group with 54 genera and at least 800 species. They're found in freshwater, saltwater.
We're gonna put slip covers on these to thin these down.
They can survive without light or on a diet of organic matter such as beef extract, peptone, acetate, ethanol, and carbohydrates.
They can eat ethanol?
Ethanol.
Acetate?
And acetate, yeah. When there's sufficient sunlight for it to feed by phototrophy, it uses chloroplasts containing the pigments of chlorophyll A and B to produce sugars by photosynthesis. In reproduction, they reproduce asexually through binary fission. So the little circular things that you were seeing were like baby,
Euglena?
baby Euglena. Yeah. In 1674, in a letter to the Royal Society, Leeuwenhoek wrote that he had collected water samples from an inland lake in which he found animalcules, which were green in the middle and before and behind white. Clifford Dobell regards it as almost certain that these were Euglena, whose peculiar arrangement of chromatophores gave the flagellate its appearance at low magnification. So there we go.
We'll move into the middle. Let's see what's going on in the middle.
They don't like the middle.
Nothing likes the middle. I mean, it's like
No, they like the edges.
They like the edge.
Maybe it's warmer at the edges. I don't know.
Maybe they know there's something outside of their universe.
They know there's something outside. They're pioneers. Yes.
Trying to go over the mountain.
Mm-hmm. So Euglena move by metaboly and swimming. Okay.
What's metabolic?
I don't know what metaboly is. That's probably how I swim, by metaboly.
I swim by trying not to drown.
You swim. Okay. Okay. Well, Kent, I think that we're going to call it a night here.
Very clearly, the edge is the place to be.
Oh, yeah. It's the hangout. Yeah, if you refocus, you'll see a lot of euglena there.
Yeah, the middle just has nothing going on.
I see something. Focus on it.
There's nothing moving.
There you go. Yes, they are. Look at them. They're all over the place. And they are moving.
And they are in the middle too.
Yeah.
Ooh,
There's something big.
Paramecium.
Yeah, Paramecium.
No, I don't think that's Paramecium.
I think it is. What do you guys think? Book Davies says that euglena water jet. And he says also that probably, and Book's good at this, okay? He says probably surface tension of the water is holding them near the edge.
You got one thing rolling around,
Yeah,
like little snails almost.
yeah. And Book's correct. He says Leeuwenhoek's drawings are amazing. The guy was an incredible illustrator. You know, if you compare his drawings to photographs made today, they're really accurate. So, okay. Well, we're going to leave you with visions of euglena,
Well, there's a lot of them.
and there is a lot of them, all in a drop of water. Okay, until next time.
See y'all.
Bye-bye.
So what are you doing with these wet slides?
I take them. Hey everybody, Scott Roberts here with Mike Hatch. Just wash
And
them.
we're here at Explore Scientific Live, and we wanted to show you the Bresser line of microscopes. Now we're just showing a few of them here. We've got microscopes that do fluorescence work, some used for metallurgy. A microscope like this can look at 3D objects, including things like circuit boards, or if you have insects or anything like that, this does an amazing job. A lot of these microscopes can also take a camera attachment, which is great.
These right here with the trinocular.
That's right, and we have a special camera that does that. Some of these other microscopes have a way to attach like your iPhone to them so that you can get images. This one right here has a built-in monitor, so it's pretty cool. The microscopic world is endlessly fascinating, just like astronomy is, and we hope that you take a look.